Dear learners, today's topic is prokaryotic expression system, advantages and challenges. So in this you will be learning first about in brief outlook of expression vectors and the differences in the regulation of gene expression in a prokaryotic cell and that of a eukaryotic cell, and how these eukaryotic genes they are expressed in bacterial cell, and you will be learning in detail about the prokaryotic expression system and the advantages and challenges occurred while expressing a eukaryotic gene on a prokaryotic cell. Before going to the actual topic, let's recap what is gene expression and what gene regulation is. So you know the central dogma of life, that is DNA conversion to RNA is called as transcription, and conversion of RNA to protein is called as translation, and gene expression is the combination of transcription and translation, which is also called as protein synthesis. Synthesis of a protein is nothing but gene expression, and it includes transcription and translation. And what is this regulation? So you are saying that whenever the gene expression is regulated means the gene expression is controlled, that is where that particular protein has to be produced and how much protein has to be produced. So that is called as gene regulation. So this regulation is done at one level in case of the prokaryotes, that is at the transcription level in case of the prokaryotes, whereas in eukaryotes the regulation is done at different levels. So let's see the differences in gene expression and regulation in a prokaryotic cell and a eukaryotic cell. So in a prokaryotic cell you will be having that is the nucleoid and a cytoplasm. So specialized membrane bound organelles are absent and the chromosome is present within this cytoplasm. Whereas if you see the eukaryote, the chromosomes are present in a membrane bound organelle called as nucleus, and also you are having separate other cell organelles are also present. So and whereas in this prokaryotes, the transcription and translation process, that is RNA and protein formation is occurring simultaneously and within the cytoplasm, but in the case of eukaryotes the transcription process is occurring in the nucleus, and also after transcription you are getting a immature RNA or heterogeneous RNA. This one is modified to give rise to a mature RNA, and that is the post transcription modification, and afterwards it enters into the cytoplasm. The translation occurs in the cytoplasm. Further in the cytoplasm, in the presence of the cell organelles like Golgi complex and endoplasmic reticulum, the protein is modified further. So post-translational modification is also occurring. So in this way, if you see the differences, the RNA transcription and translation is occurring simultaneously in the cytoplasm in case of the prokaryotic cell, but in eukaryotic cell the transcription is occurring in the nucleus but the translation is seen in the cytoplasm, and the gene expression is regulated only at the transcription level in case of the prokaryotic organism, whereas in eukaryotes the gene expression is regulated at epigenetic level and the transcriptional level, post-transcriptional, translational and post-translational level. So that is the protein is highly modified at different levels in case of the eukaryotic organisms. So these are the major differences seen in the regulation of gene expression in prokaryotes and eukaryotes. And now coming to our actual topic, that is the expression vector and how this expression vector is expressed in a prokaryotic expression system we will be learning. So you have seen that the expression vectors are having the ability to express the foreign gene because it is having regulatory elements. With the support of the regulatory elements, the inserted desired gene, it is being expressed. Okay, so the host expression system, you have learned that in order to express a protein, the expression vector must be transformed into a host cell. So these host cell, it provides the optimal environment for the growth and replication of the vector. So whenever you introduce the vector in the host, depending upon the host, that is, if it is a prokaryote, it is called as prokaryotic expression system. The example is E. coli. And if you are introducing our vector in a eukaryotic cells like yeast cells or insects infected with this baculovirus, insect cells or mammalian cells, then it is called as a eukaryotic expression system. In this session, you will be learning in detail about the prokaryotic expression system. So let's first see why this expression system is preferred. What are the advantages? Suppose the one which you are taking most prominently is the E. coli expression system, because it is having simple physiology and the entire genome is sequenced, and it is the genetics of that organism is clearly it is understood. So this is why it is most preferred. It is easy to manipulate and it is showing fast expression, and the doubling time, the generation time is very less. So E. coli cell, it will be doubling for every 20 minutes, and it is also easy to purify from the inclusion bodies. The proteins, whenever you are tagging them, so you can easily purify them. And also well established labeling protocols for stability studies is also available, and it is having a regulatory track record and easy to scale up by fermentation, and the culturing cost is also very low, and you can produce the protein at a large scale by using this E. coli cell. And the other property is it's having strong anti-pollution ability, and is a wide application range. It's a broad host range, so you can use different types of vectors can be introduced in this E. coli cell. So these are the advantages. For this reason, the E. coli expression systems are most preferred. And coming to the challenges, so what are the limitations if you see and how they are overcome? The first one is the stability of the RNA. If you take the mRNA of this that is encoded by this bacteria, they are containing less GC content, that is this nitrogen bases G and C, they are less, whereas it contains high this adenine and thymine bases are present in high ratio. So due to this the stability of the mRNA decreases. So this is one of the challenge faced. And the second one is translation efficiency. So translation efficiency, it is governed by the composition of the promoter, especially at the translation initiation site. This Shine-Dalgarno sequences, it enables the efficient binding of this ribosome. So if the distance between this SD sequence and the start codon is also very important for efficient translation. If the distance increases, the translation efficiency decreases. So that is the mostly the distance should be between 6 to 9 nucleotides. That is the distance between the SD sequence and the translation start site. It should be between 6 to 9 bases. If it is more than that, the translation efficiency it decreases. So next factor is the codon usage, codon bias or codon preference. So you know that genetic code is degenerate and there are 20 amino acids, and for that 20 amino acids 61 codons are present, and always these organisms they prefer certain types of codons. So this preference is called as codon bias or codon usage. So if the protein is more heterogeneous, so you require more tRNA to recognize the genetic code. But if the host expression system has low level of particular tRNA, or if it is not having any this particular tRNA, then it will delay the synthesis of the protein, or it stop the synthesis of a particular amino acid. So due to this, what happens means less protein is synthesized, or it results in the production of a truncated protein. So this codon bias, it also plays a major role in expressing the protein, heterologous protein in the bacterial cell. So next factor is post-translational modification. In a bacterial cell, cell organelles and these enzymes, or that is the enzymes which are required for this post-translational modification, they are absent. The organelles like endoplasmic reticulum, Golgi complex, kinases, and the enzymes which promote this glycosylation are absent in bacteria. Due to this deficiency, the bacteria are able to produce proteins which are biologically inactive, and sometimes they are producing they are affecting the structure and the function of the recombinant protein. So this is one of the challenge to be overcome. Next, coming to the other feature, inclusion bodies. So whenever a protein is overexpressed, it accumulates in the form of inclusion bodies, and they are difficult to solubilize. So the purification process is also tedious, it is time consuming, and it involves very use of this harsh chemicals, and which results in the damage and the denaturation of these recombinant proteins. And the other factor is toxicity. So whenever the particular proteins are overexpressed, it becomes toxic to the host cell, and it prevents cell division, and it decreases the biomass. And sometimes when the host cell is like this E. coli or Bacillus subtilis, if it is taken, it causes the accumulation of the endotoxins which denature our recombinant proteins. So this is also one of the challenge. The next one is protein folding. So the expressed proteins, if they are not folded properly, it affects the structure and function of the protein. So normally the proteins expressed in the bacterial cell, they are directed to 3 different locations: in the cytoplasm, periplasm, and in the growth medium through secretion. So there this non-native disulfide bonds, they are formed within this peptide chain, so which results in the formation of this insoluble aggregates, and it causes misfolding of the proteins. So this protein folding, it is also a major drawback in this prokaryotic expression system. And coming to the last one, that is the fusion tag or the purification tag. So this is the short stretch of the DNA which encodes the small peptide, and it is tagged either to the N terminal or C terminal of our desired gene. So this results in the formation of a fusion protein. So fusion protein means tag peptide plus our recombinant protein, it causes fusion protein. So this is the target protein or our recombinant protein. So when this one is expressed along with our desired protein, the affinity tag is also expressed, and you will be getting a fusion protein. So it helps in improved protein expression, stability, and resistant to this proteases, nucleases, and it increases the solubility and purification process also. The presence of this tag to our recombinant protein, it enhances the stability and purification. In this picture, let's see how the fusion tag it works. So here this is the affinity column chromatography chamber. So here you are adding the cell lysate. The cell lysate, it is containing the total proteins, that is the host proteins plus our recombinant protein. And because our recombinant protein is tagged, our protein, it gets binded to the matrix of this column, and when you add the washing buffer, all the host proteins, they get eluted or separated or washed away, and they are collected. First they are collected, that is the host proteins are collected when you use the wash buffer. So afterwards now the column is containing only our recombinant protein, because it is the fusion protein, it is tagged whenever you use specific eluents or specific chemicals, that particular tagged protein will be coming and it will be separately isolated. And when you use certain enzymes, the particular tag is cleaved away from the recombinant protein and the purified protein can be isolated. So in this manner, the fusion tag, it helps in the purification of our recombinant protein. Now let's see how a recombinant protein is expressed in a host cell in detail. The vector taken here is PET vector. The host cell is a mutant E. coli strain. So then how it is inducing the protein expression and the cell lysate preparation, the binding of recombinant protein to the column matrix, and how you are eluting the purified recombinant protein. So all these steps you will be learning. So this is the host cell, that is the mutant E. coli strain. So this is a genetically modified E. coli cell which is having majorly two changes in its genome. One is it is having the ability to encode T7 RNA polymerase enzyme, and the next one is it is the proteases of this particular host, they are inactive. Host proteases are inactive. So these are the two changes which are seen in this E. coli strain. So by taking this E. coli strain, you will be producing the recombinant protein. The vector taken here is a PET vector. In this picture, you will be learning how the mutant E. coli strain, it expresses our recombinant protein. So the vector taken here is PET vector which is having T7 promoter. The E. coli strain, it is having the integrated T7 RNA polymerase coding gene. So here you have to know 2 points. The LAC-I gene, it produces a repressor. The repressor, it represses or stops the synthesis of this T7 gene, that is the RNA polymerase, T7 RNA polymerase, whereas inducer, it induces the synthesis of T7 RNA polymerase. So in the absence of inducer, the LAC repressor protein, it binds to the promoter and it represses the synthesis of T7 RNA polymerase. When that is when this RNA polymerase is not available, the transcription and translation of this target gene is not seen, because the further synthesis of this target gene, you require T7 RNA polymerase. So in the presence of the inducer in the form of IPTG, so the repressor, it becomes inactive, and when the repressor is inactive, the this particular T7 gene, it is expression, it is in T7 gene, it is expressed to produce a T7 RNA polymerase. This polymerase goes and binds to this promoter, and the target gene, it is expressed, and our recombinant protein is produced in a large scale. In this manner, the repressor will be inactivating the protein synthesis, whereas when you add the inducer, it activates the protein synthesis. In this manner, the gene expression is regulated by means of the repressor and the inducer. So you have known that when the T7 RNA polymerase, it binds to the T7 promoter present in the expression vector, it drives the transcription and translation of our target gene, thereby producing a large amount of the recombinant protein. So once the protein is produced in a large scale, you will be harvesting the host cells, and they are lysed with the detergent or heat, and that particular recombinant protein is released into the cell lysate. So the protein may be present either in soluble form or insoluble form. If it is present in insoluble form, it is converted to soluble form by using certain types of agents like urea or detergent. So this cell lysate containing the total protein mixture, that is the host cell proteins plus our recombinant proteins when they are loaded in affinity column chromatography, and later when you use washing buffer, so all the unnecessary proteins, this is the host proteins, they are washed away, and our recombinant protein which is having the tag, it will be bounding to the specific matrix present in the column. So in this manner you can separate our recombinant protein from the host proteins, and later by using specific enzymes or chemicals you can separate the tag from our recombinant protein. Then the purity of the recombinant protein, it can be tested using SDS page or Western blot analysis, and in that way you will be getting a pure protein. So here in this particular flow diagram, the entire production process is represented. So this is the PET vector containing our target gene. When this recombinant vector, it is introduced into the host cell, later the host cells, they are multiplied, and by taking appropriate medium and by providing proper growth condition, the recombinant host cells, they will be multiplying. And when you induce it with IPTG is the inducer which helps in the expression of our protein. So once the protein is expressed, it is present in the cell lysate. Then if you centrifuge that one, the pellet, you will be cell lysate will be and the supernatant, it is separated. Then the cell lysate, if you subject it to affinity column chromatography, you will be able to get the our recombinant protein, and the purity of the recombinant protein can be checked by means of the SDS page analysis. So in this manner you can produce a recombinant protein using E. coli expression system. So in the last, let's conclude the session by just rewinding the main points. You have known that expression vectors are those which express the desired gene, and you have even known the difference in the regulation mechanism of gene expression in prokaryotic and eukaryotic cell. The two types of host expression systems you have seen, the prokaryotic and eukaryotic expression system. Of that you have seen the what are the advantages of prokaryotic expression system and what are the challenges faced in order to express a eukaryotic gene in a prokaryotic expression system. And you have seen the that is the model of E. coli expression system in producing a recombinant protein. With this I conclude the session. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.